Okay, so I want to go back. So morning we discussed this coalescent theory, which is I said it's a retrospective view of evolution. I want to come back now to prospective view. That is the sort of thing that we had discussed in the first 3 lectures. That is, again, to raise my initial condition, I want to know what happens next. So, but in the multi-loci stochastic model setting. So we have discussed things like this: single locus deterministic models, multi-loci deterministic models, single locus stochastic models. That's what we did yesterday. And I want to discuss stochastic multi-loci models. Technique is one thing, and I want to indicate what are the problems that one faces when one tries to understand these problems. But also I want to explain a couple of biological questions of interest. So in all these cases, I'll have stochastics already have random genetic drift, selection and mutation. Okay, so let me start in the following manner. So there is this broad question of evolution of genetic systems, so which means the following. So we have our genome organized in a certain way. There are that many, something called codons. There are some spaces which don't do presumably nothing much. There's a way the genome is organized. But how did it, what are the organizing principles? Do we know that? Can we understand those numbers, etc.? So how did the genetic system itself? So I have been saying, take a genetic sequence, do this, and that to it. But what about the sequence itself? Right? What is the structure there? What are the patterns there? That's a sort of broad question of evolution of genetic systems. So there are a couple of things that lots of people, but there are a couple of things that we are interested in. One is about some cases, some organisms are asexual, others are sexual. So that's the problem of evolution of sex and recombination. So let me just start with that. So I'll just basically focus on that problem first. So the specific biological problem is called evolution of sex and recombination. So the question is easy to state. So the problem is the following. So when life started, at that time, the cells just divided, they just self-replicated. They just made 2 copies of itself. Now in the second lecture, I showed you something called recombination, in which I said that, you have to mix the genome. There's a genetic mixing. As far as we know, there are millions of species. Out of them, only 400 or thereabouts, are obligate asexual. That means, from whatever we can say from, I don't know how, but we can say that, it seems, that for millions of years, they haven't had any genetic mixing at all. They have always been making 2 copies of themselves, just divide. They don't mix material, 2 of them. So they have been obligate asexual for millions of years. Okay? But rest of the species, there's millions of them, the species in the, as far as we know that, they all, at some point or the other, in the life cycle, do mix genetic material. If you think about this, why do the mixing? Because if you want to mix a material, you have to find a partner, right? The partner might have a disease, so you're taking a risk. It might also be a good one, so that if you mix, you might produce a better offspring. That's possible. So it's not obvious why you should mix genetically. Okay, so this is a very broad and open, broad question. So it's sort of, there are lots and lots of models, and it's a very intricate question, but it's quite interesting. There are several explanations, theories about why sex evolved. Why do most species reproduce sexually? Okay? So I'll give a couple of such theories and try to say that, what do they say? And when I try to make a model, what happens? What kind of technical difficulties do we land in? So I'll just describe under this big heading a couple of models. So one of them is called Muller's ratchet. Okay, it's a stochastic model I'll describe. Other one, I'm not sure what the name is. Okay, I'll just say Fisher's explanation. Okay, I'll describe these things one by one. Okay, so let's just consider Muller's ratchet first. So Muller, also a Nobel laureate, and he was one of the important persons to understand that how does mutations work, what is the effect, etc. So he got a Nobel for that. Okay, so but this is a sort of simple verbal model that he gave, and later on it was worked out by Muller, no, not by Muller, by Haig. Okay, so the model is this. So we think of n asexual haploids. And I have a certain fitness scheme which I'll specify. And the idea is that I have this n of these guys, and then then one is thinking of, so let me just specify that a bit more. So one is thinking that I have some fitness landscape. So see, remember I said selection and mutation. And I'm now thinking of multiloci. And on second lecture, I spent some time talking about these fitness landscapes. So I said they come in various shapes. So one of them shape is that Mount Fujiyama, in which there's a single peak. So there's all the ones at the constant radius have the same fitness, and it reduces till it goes the peak where it is 1. It's some number, constant number. Okay? So it's something like this. It's just a cartoon. Okay? So this is the sort of fitness landscape. And imagine the population, all n of them are sitting right at the top. So all the organisms have fitness 1 to start with. But then, because it's a finite population, there will be stochastic fluctuations. So let's imagine this model of n haploids. This is the fitness landscape, and there are mutations. And I just run a Wright-Fisher model. So this is the selection part. That's the genetic drift part. And mutations are like this. Mutations are such that the good can become bad, but this we ignore at a certain rate. And the logic behind this being that most mutations are deleterious. That means if I am at the top, it's more likely that I'll get worse. My car, remember, mileage would go down. It's unlikely that by just random changes it would become better. So that's the sort of logic. As a zeroth order approximation, these numbers are much smaller than these numbers. Okay? So what do you think would happen? So I'm starting with a population, n of them, all have 0 mutations, 0 deleterious mutations. And I run this Wright-Fisher process, in which the next generation is formed by choosing a parent with a probability proportional to its probability proportional to fitness of the parent. And I run this. So there's a one-way mutation, and it's a genetic drift. What do you think would happen after some time? So everybody has 0 mutations. Let me say I denote. Let's say n is 4. Each of them has 0 mutation. 0 can become 1. It can acquire a deleterious mutation. Good can become bad. Okay, I'm not allowing the back mutation. What would happen? Not all, because some would become 1, some can become 2, etc. So there's some lot of time has gone inside this. So I'm just plotting this, I'm just drawing this, and after a long time, this has happened, and so on. So what is going on? So this is this is sort of obvious, because that mutation is one-sided. You might think, I'm saying it's obvious, but if you have an infinitely large population, then the answer is different. So I haven't told the answer, so sorry, let me step back. Okay, so I'm saying if I have a finite population, something like this would happen. So what the meaning of this is, that I have lost the population with 0 deleterious mutations. Everybody has at least 1 bad mutation. After some time, everybody has at least 2 bad mutations. And because I'm allowing only unidirectional mutations, it's a ratchet. That's why this word ratchet here. And because of Mala ratchet. So the point here is this. So if you look at this such a model, if you simulate it or set up a certain theory for this, then the argument was then asexual population will degenerate in time and therefore should have genetic mix. So this is one argument that in a finite, it's very important to stress this point, that it has to be a finite population. If I had an infinitely large population, so I had sort of indicated to you to work out an exercise, but let me just remind you that if I had n infinity, then the fraction of population with zero deleterious mutations is non-zero, even though I have unidirectional mutation. So this is the statement. If there's no genetic drift, fraction with zero deleterious mutations is non-zero, although I have one-way mutation. This is something you have to work out. Okay, it can be done quite easily. You just have to set up this model I had written down on I think second or third class. But n is finite. This becomes 0 after a certain time. So this time is the click of the ratchet, and people are interested in calculating this time. Okay, so there have been various ways of setting it up. For example, you can set up a Fokker-Planck equation. Right? You can think of that, so that sort of Kolmogorov equation we wrote down. But one locus, two allele model, I had just one variable, the frequency of A1 allele, right? So the distribution was of just single frequency. But now I have 0 type, 1 type, 2 type, 3 type, and so on, right? I have x0, x1, x2, x3. This is a higher dimension. It's not a one-dimensional PD. I mean, it's 1 plus 1, what we did yesterday. It's going to be 1 plus lots of space variables. Those things we can't really handle. So I'm trying to say, so I just gave you the argument. What I'm trying to say here is, if you set it up the way we did for single locus models, it's extremely hard to make analytical progress. I want to show one more way of doing this thing, and will remind you of something that you saw already, I think, in Martin's talk. So let's just say that, fine, maybe we can do something. Let's try to set up a little equation for the cumulants or for the moments and see what happens for this model. So my aim of doing all this is just to indicate to you that if you want to do these kind of problems, that's where the action is. Much of the research is focused on these kind of problems, multi-loci stochastic models. There are problems when you try to do things analytically. Numerically, of course, you can put it in a computer. But just to know like what kind of problems are very similar to what we encounter in our StatMap problems. Let me just demonstrate it for this case. Okay. So I'm interested in writing an equation for the average number of deleterious mutations at time t plus 1. Average, where I have done a stochastic average. So there are two types of averages I'm writing here. This refers to population average. That is, I go to the first guy and ask, how many mutations do you have? 1? Okay, how many you? So 1 plus 1 plus 2 plus 2 by 4. That's what I mean by these angular brackets. And these bar means the averaging over histories, stochastic histories. Okay. So just keep that in mind. So I want to write an equation for this guy. So suppose I had at generation T, I had, let's say, these individuals. Let me see what example I wrote. So at some generation T, I had these four individuals. First individual had K1 mutations, second K2, third K3, and fourth also has the K2. K1, K2, K3 realize anything between 0 and infinity. Okay? The next generation, let's say this got K2. Yeah, so we are assuming that my genome is very, very long. So I'm assuming L to be infinite. It just helps things because we discussed the scaling limit in one of the lectures, in second lecture, I think. So I said that these models simplify when I take L to infinity and the mutation probability to be 0. I think it was the same body. So that's that kind of limit I'm assuming that it has been taken here. You can restrict it if you want. I mean, that's also fine. There are models with that also in which you say this many mutations, and after that, I'm actually going to kill the guys because they are so unfit. Somebody, if it reaches here, I'm just going to start killing. And then if you are reached a maximum, you're dead. You are dead here itself. Some threshold you can put. Okay, so this is the Wright-Fisher, and on top of it, I could put mutations, which is chosen from some Poisson distribution. Okay, so that's not very important, but let's just put it anyway. Okay, so so I've written down this, the first guy, the first offspring had some K1 prime mutations. So K1 prime here is K2, K2 prime is K2, K3 prime is K3, and K4 prime is K2. It's just a mutation. And this is the mutations. So this is just copying the genome, and then on top of it, you could have mutations. Okay? So, and there's an average here. And mutations are independent of whatever you have inherited. So this is sort of unimportant, and I'll just write on an average is some number like mu. Let's just find, okay? And I want to really deal with this guy. So how many mutations I got here? Let me write it first. K times the average this term would give me 2K2 plus K3 plus K2. So this guy produced 2 offspring, right? So I would be writing here K2 into 2, K3 into 1, and K2 into 1. So K, the number of mutations, and average number of the offspring, which is like 2 here in this particular example, right? And because I want to average it over lots of stochastic history, so this is one history. I could do another one. Some other realization, I'll get different numbers. So that's what this average is over. Okay? Just to think a bit, it will work out. Okay? So this one we know. How do we know this? From yesterday's lecture? Average number of offspring produced per generation by an individual of type K in Wright-Fisher process. It was binomially distributed. I want to go through this a little hurriedly, simply because I want to make just a technical point here. Okay? So this one is, again, binomially distributed, as we saw yesterday. And therefore, this is parent times average NK. And for that fitness scheme, I'm just giving you the answer. If you don't get it, feel free to write to me. This is Ki, and so what I've done is just use the fact that I had average NK, which was n times the probability proportional to fitness. This probability, maybe I'll just write it. So which is equal to average fitness proportional to the fitness. And this fitness for that case is 1 minus s to the power k, and this is this. Okay? You just take it right now. And if I plug it in, over there. But the point here is now the following. If I now do this sum, I get something like this. Plus there was this mutation. average number of mutations at time t plus 1 is equal to whatever was there previously, and this new guy and the mutations. Does it remind you of something that you saw in one of these lectures this week? So we're writing the equation for this guy. For this model, it turns out to be this. Is this a good news? Huh? No, M is not important. Let's forget it. That's not an important part. I'm writing equation for average k at t plus 1. Is this a good thing or bad thing? Good? There is the recurrence relation, but there's a big but. Equations are not closing. You already saw that in exclusion process, right? I have this object for mean. This is mean at time t. That's good. This guy is a variance. So M1, the first moment, is related to the second one. Okay, so this is the same kind of problem. So I just used this model just to demonstrate this little point. When we try to do the same technology when we use, we wind up in the same problems. Again, when we write these equations, there is a hierarchy. I write equation for M2, it has M3, covariances, etc., and it's just not possible to solve it exactly. So this model is very simple, but to date, it has not been solved exactly. Very simple to state, actually. Okay, so some diffusion equations, etc., we can write. This is something we have worked on. You get some reasonable answers, but it works sometimes in some parameter regime, otherwise not. But something like this, because because you have a hierarchy, we tend to close it by hand or by some input from simulations, etc. So the same kind of ideas are used in this subject as well. Okay? Yeah. Okay, so I already said if you write Walker-Planck equation, it's higher dimensional. Again, that's a problem. You can't really solve it. Okay, so that was the biological question. That was the motivating question, that why sex in recombination? So one argument being that asexual populations are finite, as all populations are. They would, in time, degenerate. They would degrade because they'll keep accumulating deleterious mutations. So therefore, there should be genetic mixing. Okay, this is one argument. I want to do the second one. Is this okay, roughly? I think if you find this interesting, I suggest that you just run a Wright-Fisher process simulation tonight. Many of these things become clear when you just show some simulations, you see, and see trajectories going up and down or whatever they're doing. It's really worth the trouble. Okay. So the other argument, so this was based on deleterious mutations. The argument of Fisher is somewhat different. It says the following. It says, okay, fine, most mutations are deleterious, that's true. But now imagine a beneficial mutant arises in, so I have an asexual population, I have a sexual population here, and most mutations are deleterious, but once in a while, a beneficial mutant will come. If it's an asexual population, and because mutations are deleterious, it already has very low fitness, even if beneficial mutant comes, the chances that this beneficial mutation can spread are small. This guy, the good guy, it's stuck in this bad background forever. And therefore, it will just keep reducing its fitness, and it's unlikely that it could spread. But for sexual populations, suppose I have something like 3 bad mutations, and something like this, let's say. This has 4 bad mutations. Okay? Let's say it was 5. And by chance, this became 0. It became deleted, it became a beneficial mutation. Then by recombination, if it happens to the correct side, if I swap it, I could generate a mutant like this, which is better than both of them. So by sexual reproduction, by recombination, it's possible to generate better mutants. Of course, there's other one also, right? But that would die out because it has a lower fitness. So that way you could generate them, and you expect that these will have a higher probability of fixing. So yesterday I said that some of us are in this business of calculating fixation probabilities. This is one of the questions. Lots of people calculate these things. So if I want to answer this question, I would like to calculate the probability of fixation of a beneficial mutant in asexual population and a sexual population, compare them and see that if indeed I asex is smaller than I sex. So that would be also an argument why genetic mixing. Okay? Okay. So now we have learned already one method of calculating this fixation probabilities, which was this yesterday using this backward Kolmogorov equations. I want to demonstrate one more method. Also, I think because it has not been discussed in this course. So this is called branching process. Have you heard of this before? Branching process? Okay, so let's do it. I'll do it for a one locus model, and then I'll just write down the final answer for the multiloci models. Okay. So the standard reference for this is a book. So I'm just thinking of again a single locus problem, and there are A1s and A2s. So let me say that I have a population of A2. Okay, lots of them. So I'm thinking there are lots of them. I am assuming population size is large but finite. Okay, so this is the approximation that's going in here. So the Kolmogorov equation, we said there's a finite population. I did scaling of time, etc. with n. Here I'm assuming that there's a large but finite population. That's one important assumption. I mean, it's a very large population. Okay. And let's say now a mutant of type A1 arises in this population by mutations. And I want to know what's the chance that eventually it would spread. Okay, eventually what is the probability that it would take over, the population would spread to the population. I want to write down a recursion relation for this. And this is nothing special to genetics or something. It's something you probably have seen in physics literature also. When you again discuss absorbing state transitions, for example, these transitions. Okay, so let me go through this. This is the probability of extinction. Fixation probability will be how much then? 1? Huh? Minus. Minus pi t. It's easier to write these things in terms of E, extinction probabilities, and we can of course subtract that and get this for pi. Okay, so this is the question that we want to address. So let's say T generation. So I'm not writing that background, guys. I want to write only on A1. So I had this copy of A1 at time T. Let's say T plus 1. Let me Let's stick with might only. This is a rare mutant. It didn't produce any offspring. Finished. Right? So this is one possibility. There was a single copy. It produced one offspring, and that did whatever, 2 offspring, or maybe it died or whatever. Okay, that's one possibility. Maybe it produced 2 offsprings. And each of them did their thing. Okay? So the thing is, I want to write a recursion relation for that probability at time t plus 1 in terms of time at t. That's why I'm writing dot, 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 dot. Let's try to write that. So that Et is equal to probability. So it got extinct, the copy present at time t, it got eventually extinct if it produced 0 offspring. So this is the probability that A1 produced n offspring. That's the distribution here, P0. Then we said this could happen. Okay, so it produced 1 offspring, and from there on, the process repeats. This is what you do. So this is the 1 copy present. Then you can ask the same the copy present at time t plus 1 goes extinct. So this is Et plus 1. Is this clear? Right? Plus, what should I write here? P2 Et plus 1 square. Both of the lineages should go extinct. Right? So I'm assuming, there's an important assumption going in, that the lineage of this guy and these guys, they don't interfere. So that's the assumption going in, and it's generally fine. It's not a problem. They don't, affect each other. Plus and so on. So you see the pattern now. This is nothing but summation. Right. And if I say that at large times where this probability becomes time independent, I have E okay. So we want to analyze this equation. This is the eventual probability of extinction. This is a probability. So summation Pn this one. So first thing is, so this is our equation. First thing is that E equal to 1 is solution of this equation. Right? I mean, first of all, it's a nonlinear equation, right? E square, E cube, till infinity, right? Okay, E equal to 1 is a solution. That means that A1 got extinct with probability 1. Are there other solutions? So I think, yeah. So let's see the other one. Let me take derivative of this equation on both sides by E. So I have okay? This is a derivative, right? If E is equal to 1, then what do I have? Okay, so let me just, let's not do that. Okay, so I want to solve for this one. So I want to solve for this graphically. So let me just see what I want to say. So this is G of E. Okay, so this is what I want to do. Let me just show that. G of 1 is the average number of offspring, right? It's mu. So let me just do this, okay, and I'll explain. So I want to plot the following for mu larger than 1. So I want to solve this equation, G of E equal to 1, and I want to do it graphically. Okay, there's a standard way of doing this. So this is what I want to do. So E lies between 0 and 1, it's a probability. Right? And so that's why, yeah, so G of 1 is mu, the number of average offspring. So the value at mu is somewhere here. What is G of 0? G of 0 is P1. Okay. This is mu. And I know already that I should distinguish between the two cases, mu larger than 1 and mu less than 1. And somewhere here I have a line for mu equal to 1. So now this is the sum of positive numbers. So G of E must be increasing function of E. So let's go like this. The solution that I want is the one for which G of E is 1. So this is my solution. Is this clear what I'm saying? So I want to solve this equation graphically, right? So G of E looks like this because I know the two limits. G of 0 is P1, right? And G of 1 is mu, the average number. And I'm just in a monotonical increasing function and just plot it out. And my solution is when G of E is equal to 1. So that's the solution. So the non-trivial solution that I have found is this E star. So I'm saying when mu is larger than 1, I have two solutions. E equal to 1 and E star. When mu is larger than 1. What happens when mu less than 1? Could you plot this? Less than 1 instead. Something like this? No solution. Why? Yeah, because of probability. So now I'm getting an intersection point beyond 1, right? So therefore there's no solution when mu is less than 1. Only solution when mu is less than 1 is E equal to 1. So what does it mean? So E equal to 1, what is this all E business? So the point is just this. When mu, so this little analysis if you do, you'll see that if the average number of offspring produced per generation, okay, if it is greater than 1, then the population can go extinct or it can get fixed. And it turns out if you do stability analysis, that this is the stable fixed point. In this case, E equal to 1 is the only one. Okay, if the number of offspring produces less than 1, average number, then it definitely goes extinct. Otherwise, there is a certain probability. Yeah? Mu equal to 1. Mu equal to 1 we didn't even try right now. So that's something in between, right? Mu equal to 1, yes. Yeah. No. The solution is this guy, when this function intersects 1. Yes, that's it. Yeah, G of 1 is mu. That's not a solution. It's not a solution, I'm saying. Solution is this guy. Okay? Okay, so what is it good for? So why are we doing all this thing? It's standard. I mean, this you can use. But yesterday we learned something else. Is this different, same? If they're same, when are they same? So let me just try to address that. So this is a small calculation. And this is some half-page thing that you should definitely try. But now let me make contact with what you said you did yesterday. So now if I choose, so I know that A1 must have a fitness. It should have, so if I choose mu to be 1 plus S, where S is positive, if the number of offspring produced by A1 on an average has to be larger than 1 if it wants to get fixed, right? So the mu must be 1 plus S, where S is positive. It's a beneficial mutation, which makes sense. That means within branching process, if I had chosen S to be negative, then branching process would tell me that for sure it does not get fixed. Is this consistent with what we did yesterday? Deleterious mutations, do they definitely go extinct? We drew some funny curve, remember? So I said yesterday is a famous formula. If I have a beneficial mutation, it doesn't necessarily get fixed. It doesn't get fixed with probability 1. There's a certain chance for it. And deleterious mutations can also spread. There's a small, exponentially small chance, but they can get fixed. Okay, that's what we did yesterday when we had the population size n. Here I'm assuming the branching process works in the case when this is a scaling factor we had yesterday, when this is much, much larger than 1. So I'm saying that this Fokker-Planck approach answer and the branching process answer, they match when the population size times the advantage when it's much, much larger than 1. Then the two answers coincide. Actually, they don't coincide. They are close by. Okay? So branching process, I find it very intuitive in the sense that I don't have to worry about calculating this Taylor's expansion of A of x, B of x, etc., etc. It's sort of intuitive to write it down. But it works only for advantageous mutations. If I'm dealing with deleterious mutations, branching process doesn't work. It doesn't capture this part. Okay? So if you're interested only in this part, branching process is a good point to start. Okay, so let's just plug this in, and I say that my Pn, if it's a Poisson process, mu mean mu, and I plug it in over here and do some simple simplification. If I do, I'll get this nonlinear equation for the fixation probability. This is exact when ns is much, much larger than 1. So if you were to do Wright-Fisher process for very large populations, you would see that this solution, this is a transcendental equation, right? For pi, 1 minus pi here and exponential here, right? It's not easy to solve. You can do it graphically or put it in Mathematica or MATLAB, whatever you like. Okay, so this gives you the exact answer for branching, for the fixation probability, provided n is large enough. Okay, so let's just finish this story. And if I put now mu to be 1, so let's compare. These are the kind of answers we had. So I'm saying if you look at ns much, much larger than 1, this term goes, s is positive, and little s is small. Okay. Let's see it. I don't think I. So this is what you will get from what we discussed yesterday. For large n, this is the answer. For small s, this reduces to this guy. Yesterday, when I was plotting this curve, I said, how does it look? And I drew something linear over here. So that's this answer. This was 1 by 2n, and then there was 2s, and then it saturates. What about this? This I said is a transcendental equation, right? But if s is small, can we solve it? Can we try? So we expect that if s is small, I have a slight selective advantage. So this will be the probability of fixing will also be small. So pi is small, s is small. Use that fact, do a Taylor series expansion, and tell me what you get. We take log on both sides. What is this? Right? Pi equal to 0 is a solution. That means that's the solution. E equal to 1. Pi equal to 0, right? That's there, of course. So, and it's a linear equation in pi, and you can see that it's nothing but so if you were to do a large n simulation, these two are going to match over here, but your answers would match with this equation, the simulations, then Kimura's. This is branching process. Let's say Fokker-Planck. So your simulations will match the yellow answer. This is exact, so long as your population size is large. Okay? So there was no explicit n, etc., but basically one could set up a calculation like this. It's not difficult to set it up, but making approximations, devising analytical answers, that's not easy here either. Okay, so I just want to write just the expression I'll write down. This was a single locus problem, analog of what we did yesterday using Fokker-Planck approach. I just write down the expression when you have a multiloci problem. I just write it down. I won't analyze it or anything. People have done that for this problem of section recombination, and basically we don't have analytical answers. If anybody has ideas, it would be really nice to work it. So in this case, if I have several types, then the fixation probability in the background of I types is of this form. So I'm not explaining all this, but I just want to give you an idea about it can be generalized. And if I make some assumption like Poisson distribution for the offspring, it's writable as so point is, again you have this sort of structure, similar structure, exponential of something. Something like this still comes. Okay, but there are several more steps that you have to do. For this multiloci setting, you can generalize the branching process, but it's a bit more involved, and there's one more step which I haven't described, but you'll have to do that. And here's the reference if you're interested. So these guys analyzed this multiloci branching process, keeping that sort of model, sex versus asex, and they indeed find that pi asex is smaller than pi sex. Okay, but it's numerical, etc. It's fine, but one would like a simple answer like 2s. So, I don't know, it can be done. Okay. Okay, I think I'll just use slides, please. So I'll just switch to slides because I want to describe one more problem. It was easier to just do it on the slides. Actually, I want to use board also. Is that okay? Yeah, so let me light this. Yeah, that's okay. Yeah. Okay, so this just, sorry. This is sort of thing you already know, so it's not what I want to say. Okay. Okay, so I want to describe one more problem, multiloci, stochastic, etc., and what's the biological problem. So I want to sort of describe that a bit here. So the question that I have in mind now is not about this evolution of genetic systems, but adaptation. So the question is just this. So you start with bacteria. So as I mentioned the other day, Lenski has been doing this experiment since late 1980s, in which he has started with these 12 replicate populations. That means he has these 12 petri dishes. Each of them started with the same, exactly the same cell, okay, same genome. Each of the cells the starting population had. And then they have been given just less food, and then they have just been trying to adapt to this bad environment. They have been doing this for, more than 25 years now. Okay? So that's the question of adaptation. How does the adaptation proceed? What are the dynamics of this process, etc.? Okay, so that's a big problem, and there is a lot of progress in that. I want to look at a small part of that. Okay? So, again, I have genetic drift, I have mutation, I have selection, and I want to look at what is called a weak mutation regime. So it's something like this. I sort of indicated yesterday, but let me just describe it now. So what I have is this. So I'm just running, let me just say in terms of simulation. So I have just n, population size n, and I'm just running the simulation, but I allow the mutations to be very, very weak. Once in a while, a mutation occurs, okay? And it's possible, okay, so let's say this big mutation, yeah? And I'm just plotting, I have this, the number of loci I have is 15. So I have 2 to the power 15, I'm thinking of binary sequences. So I have 2 to the power 15 such sequences, and this is the size of the population, and mutation probability is very low, just 10 to the power minus 7, very tiny. So what would happen? I started with population, forget these numbers, they have a meaning, but let's just look at these numbers instead. 0, 0, 0. The dash, dash, dash again represents things which are same in all these things, so that's why I'm not writing them. So 0, 0, 0. The fraction of that population was 1 for a long time. And then a single mutation happened here, okay? And it was better. So that's what this number is, lower. So I have rank ordered them. The good guys have a lower number associated with them. The rank is, so these are in the, these are fitter ones. Okay? So this guy, the blue guy, it was fitter, that arose, and it acquired a frequency of almost 1. That hanged around for a while and went away because a mutation happened here, and that purple one came up, stayed for thousands of generations, went away, and then this light blue and so on. Okay? So the reason I have 15 loci, okay? What I have done is to choose this regime in which basically population is sitting at one sequence most of the time. At any point, there are just 2 types present. That's helpful because then I can appeal to single locus problems, which we discussed yesterday, and try to make some progress in this. So we'll ask different questions here. Okay? If I increase the mutation, it's a bit more complicated. So again, the red ones, and they got lost. Mutation happened here. So it rose, it didn't really stay at frequency 1. And because the blue one, while this one was going up, there is a better one, blue one arose, and tried to go up. But when it sort of tried to stay put, yet another one came. And that was so much better that it stayed for a quite long time. But then that also went away and this red one took over. So there is a lot of competition going on at short times. And at long times, again, it's as if there are just 2 times, even though the mutation probabilities are pretty high. Okay? So you might object that why I'm taking such a mutation probability to be low. But the point I want to make is it's a simple model that one can cook up. And it's not my model, it's in the literature. It's sort of something people introduced long time ago. But there was not enough progress. So I'll try to describe our results for that. So selection is strong here. Yes, selection is strong. Yeah. Yes, it's quite strong in this one. Yes. Yeah. So only mutation I'm changing, just so that you don't feel that, oh, why is it so low? I mean, the thing is that later on, you just wind up with this kind of 2 types present at any time. So one simple model is this. We studied single locus models. We spent a lot of time saying, this is just one type, and so on. What is the use of that? Nobody has a single nucleotide long genome. But it's the same physics attitude. Start with something simple and then build upon it. Okay? So those don't throw them away because it's too simple or something. It's useful. Okay? So here I have multiloci problem, but at least in some parameter regime. In this case, at short times, and in this case, at long times, I have just 2 types, and I could appeal to the results that are known. Okay. This problem is particularly interesting on rugged fitness landscapes. So that's again something that I had introduced. So let's recall, smooth fitness landscapes are these single peaked ones, in which wherever you start, you take mutations, you can always go uphill without ever going down. You can always find a path from wherever you start. That's the first case. That's the smooth fitness landscape. Rugged ones are like Himalayas. You start, you reach a local fitness peak. In physics, we call that local energy minimum. You get stuck there in the sense that you will sit there, but then if you want to go to a higher peak, you'll have to go through the valley and then go up, right? Okay. So on rugged fitness landscapes, this is sort of interesting, but the population can get stuck. So it started from here, it could reach here, it could reach here, but then, mutation probability is low. If it wants to get out, it has to acquire several mutations. Mu square, mu cube, but mu is small. It's going to take a very, very long time, right? So it has to produce some mutants like this and then go up. So that's a time much longer. So let me just ask the following question. So I'm not interested in asking, how do you go? Suppose you go here, and then you go here. So those time scales, let me not work on. Let me just ask, if I start from here, what happens just along this path? And remember, I'm just drawing it as in 2D, but actually, the back of my mind, I have the Hamming space. It's just a cartoon. Okay, so it's not like just a simple linear function, exponential function. Actually, I have the Hamming space. That's where the evolution is happening. Okay, yeah, so this is the Hamming space. So I'll just define this model. As I said, it's not some physicist model, and this is actually by biologists. And it's a sort of nice model, which works in the limits that I said. Mutation is weak, so that basically you can think of only one sequence occupied at any point of time. There are some times when both are, but that time scale is negligible. So that was Wright-Fisher, and now we can simplify things and write down a model of this kind. So this is called adaptive walk model. So let me explain that. So I have taken here a sequence of size 4. So this is 1, and I am working on rugged fitness landscape. So how do I generate them in a simulation? So what do? I just take some distribution. We'll have to worry about that later. I take some distribution, and I go to each sequence, 2 to the power L sequences. I take a random number and just drop it. That's my one fitness landscape. I can do the averaging over the fitness landscapes also if I want. Okay, so that's what this bar represents. Lighter color, low fitness. Darker color is high fitness. So I start with some moderately fit sequence. has 4 neighbors, this guy, this guy, this guy, this guy. What it does is, it wants to go uphill. It's adaptation, that means going up. So, but mutation is low, so it can't look at all the other guys. It looks only at the nearest neighbors and sees, aha, this has a lower color, a lower fitness. I'm not going to go there. Okay, it doesn't go there. And then it looks at these 3 guys. All 3 are better. It decides. How it decides, we'll talk about that. It decides to go to, let's say, this guy, 0010. And then the process repeats. It again looks at its nearest neighbors. This guy, it can't go back because it's first. And then goes up, and let's say it reaches here. This is a local fitness peak. That means if you go away from here, like this one, or like this, or here, fitness is reducing. Global fitness peak is somewhere here. The population is sort of going to sit here. And I want to understand how many steps does it take to reach a local fitness peak. That's the sort of question that we have in mind. So you're starting with some, antibiotic, a bacteria, and it has been given some antibiotic. It's in a bad environment. Then how much time would it take for it to get adjusted to this new antibiotic? How many mutations it would have acquired? How much time would it take for it to develop resistance to this antibiotic? That's the sort of question that we have in mind. Okay? And of course, this guy could have just gone directly here, or it could have gone, not to this local, it could have gone like this. Okay? So this is sort of model. Okay. So I haven't specified how these neighbors are chosen. There are several ways one could do that. And the reason I'm doing just this bit is simply because it's a problem that's appealing to physicists. Okay? So one could choose it, what is called a greedy walk. So greedy walk is the following. So what one does is, this guy, it chooses a neighbor, it looks at the 4 neighbors and chooses the best of them. So I wanted to say something a bit here. Yeah. Okay. So that in statistics is known as recall. So let me explain the problem. So that's adaptive walk and stuff, okay? So this problem, if you just think about it a bit, I'm saying that you start with a sequence of type 0, okay? You look in your neighborhood, that is, so if you have a sequence of size 4, you just make a single mutation. How many mutants would you have? If you just make a single change. Huh? L. If L is 4, yeah, 4. So you just look in your neighborhood, there are L such mutants, and you choose the best of them. So let's say the initial fitness is some value. Okay, let's just call it 0. This is step 1. So the probability that would take the first step, yes, so it's not fitness. Probability the first step would be taken would be how much? Okay, I'm appealing to some result from extreme value theory. It says that all of them will be equal. So let's say all of them are equally likely to be chosen. Okay? Because right now I'm starting with 0. So they all are better because fitness is positive. So they're all equally likely to be chosen. And the L of them. So this fitness would be, probability is 1. Then for 2, it's L into 1 by 2. So let me just explain this bit. So I'm saying here, so you've chosen one guy. So it has gone to, let's say, this one. Okay? But this one would go look in its neighborhood and it has to go to something which is the best amongst those L. That means it's going to somebody, so the one that is going to is the best amongst 12 random variables. So this one was already better than L. The neighborhood of this is another L. So when this goes to the best among this guy is the best amongst 12 random variables. Okay? Okay, I don't know. Okay, so this is 12, okay? And it could have been any of them, etc. So this is half. So this is simple, but it's a bit tiring even for me. So I'll just write it down. So probability, if you try this way, it turns out to be probability that at least k steps are taken. Turns out to be 1 by k factorial. And therefore, probability exactly k steps. So I mean, this is 1 by 2, and if you try the same way, this would be 3 factorial. Just have to repeat this argument a bit, okay? I can discuss it later if you're interested, but let me just write this down right now. So this turns out to be, this just follows from here. And this is the basic point I wanted to make, which is the following. For the average steps, so the question I'm asking here is this. I want to reach the local peak, and then I'm just stopping the game. So what do? I start with something, I look for the best amongst my neighbors, go to that. Then look at the best amongst that new neighbor, and so on. What do you think? Would the walk keep going on? So this is called a walk, adaptive walk. How much do you think steps could be taken? 1 step, 100 steps? Some intuition you have for that? Would it be pretty soon, or very late? Yeah, so it's some rugged fitness landscape, very rugged, and I'm not asking for a number, but just, some feeling for it. Actually, it's just, okay, so I'll just tell you for this case. It just turns out to be E minus 1. So the average number of steps you would take before you get stuck in a local fitness maximum is just 1.7. Because you exhaust the beneficial mutants very, very fast. You're going to the best one, then the best one, then the best one, okay? It's, you just exhaust the good ones very soon. This is certainly true for maximally rugged. That means when you choose these random variables to be totally uncorrelated ones. Not correlated. No, peaks we are not going to cross at all. No, sorry. These peaks and all don't enter the picture at all. Because we are going to get stuck here. So we start from here, and we are going to go here, and that's it. No, this is the local peak. Okay, so that's the number of steps. So that turns out to be just 1.7. I don't know if it correlated. I'll have to look up, I'm not sure. Okay? Okay. This is one walk. So I just want to give sort of a limits for these things. So if you just choose always the best one. And if you try an algorithm in which you just look at a suite of mutants which are better than you, but you can go to any of them. So you don't care whether it's the best one. You just buy anything which is better than the current pencil box you have, right? With equal probability. So that's called, so I just exactly write it just here. That's called random adaptive. So this is OR. So he writes very well. If you get some time, try to read his articles. He writes extremely well. Okay, so the other one, limit is random adaptive walk. In which you choose a better one, but all better ones are equally likely to be chosen. So So this was done first by two physicists, Flyvbjerg and Lautrup. Okay, so let me just write this recursion relation. So, these guys wrote a recursion relation for this quantity. It's saying, so this is not difficult. So let me just try to explain this bit. So it's probability that at the jth step, the fitness is H. So they write down recursion relation for this object. So the probability that the fitness is F, and the next step is that it was H in the previous step. I have at least one beta mutant available. To the probability the transition occurs. This is not difficult, okay? So it's just saying, start with this, you have better ones, and then you make a transition. Okay? So in this case, this transition probability for random adaptive walk is pretty simple. It's just equally likely. So it turns out to be something like, okay, it's just a very simple one. Okay, so I think I just saying too much right now. So let me just try to sort of wind up things. Okay, so let me just put up on it. Suppose I chose these fitnesses from a certain distribution P of F. Then this function is simple for random adaptive walk. But you're choosing things equally likely. You can look up this function, it's fine. But the main point I want to make here is neither the greedy walk nor random adaptive walk are biologically realistic. What is realistic? What is this transition probability is what we learned yesterday. So the probability that if today population has a fitness H, tomorrow it has a fitness F, when this little intersection happens, when the new population takes over, this probability is given by this object. This is the object that we calculated yesterday, 1 minus e to the minus 2s. That's the sort of bit I wanted to, the point I wanted to really make. So you have a somewhat complicated recursion relation. You can solve for this PJH only in a one special case. We could do only for, and this P of F is exponential. We could do a bit more in generating function. We could write for this and do a bit more. But explicit answer we could do only for exponential case. But if you plug it in here and work it out, it is possible to know the analog of how many steps does it take if the population is following the biological realistic situation. So this is a simple question. If I choose the best, I exhaust the beneficial mutations in E minus 1 steps. If I am happy with just going to the better one, then the answer is you reach a local fitness in log L steps. So for greedy, it's E minus 1. For random adaptive walk, it's log L. The question is what happens here? Okay, that's something that we did, and I think I'll just try to wind this up. So this is the case. So this log L, you can cook up a simple argument for this. But when you do this guy, then this is what I was trying to show you. So we find that there is a transition in the average number of steps taken. There is a certain quantity which I didn't explain, and I just don't want to right now. If the distribution, the P of F, if it has a finite mean, then again the average number of steps is like log L with a certain prefactor which you have to calculate, which we could do. But if the mean is not finite, then it's a constant. So it sort of interpolates between the greedy walk limit and random adaptive walk. But the slopes, etc., you have to sort of work it out. And this is something we could do. Okay. Okay, so I'll just, I think, try to finish this. I haven't told you that all that I wanted to, but I think it's just getting too much even for me. Okay, so this is the last bit. I calculated this walk length and all that. It's a nice physics question. How many steps does it take to get stuck at a local fitness peak? Is it relevant for experiments? Yes, experiments measure this. Okay, so this one is an experiment on some yeast. And what these guys did was to just let their population evolve, set their population trajectories for different plates. And then they see that the fitness is sort of saturating. And it's possible to figure out how many substitutions happened till the fitness reached a plateau. The plateau reaching meaning it got stuck at the fitness peak. And they're starting from here and figure out how many adaptive substitutions happen. Okay, so this is their data, mean walk length, and they find it's a constant. So we said no, you're wrong, but they're saying no, okay, I don't know. Okay, so something like that, fine. So this is the experimental answer in one experiment, but there are other answers like in viruses where there is more structure. Okay, so this is fine. I think I should just stop here. I'm just running, my birds are running faster than me. So I think I'll just, can I just stop here? Thank you very much. Sorry, I'm not able to focus anymore. Yeah, I think I'll just stop here. Okay? Yeah, one second. I wanted to show this picture. So this is what I was saying. When you measure fitness, this is a bacterial colony, bacterial lawn. So this is from an actual experiment of this person, Burch. So she has this bacterial lawn and she dropped these virus particles and they're just growing in these concentric circles. And she measured the area as a function of time. And she saw in one case, fitness increased. In the case, it remained almost a constant. It was decreasing a bit. So the interpretation is that this guy is like this. This one is perhaps sitting already near the fitness peak. So this is the sort of structure of the topology of the fitness landscape you could infer by experiments. And this was the other experiment that I mentioned where they generated lots of mutants and measured the fitness and drew a sort of a graph. And in that case, it turned out to be smooth one. So smooth fitness landscapes is Mount Fujiyama, which is used a lot in theory. They are experimentally seen, and there are also these rugged ones. Okay, so these are sort of very old experiments, but there are lots and lots of them that you could look up. Okay, this time really stopping. Sorry? Yeah, so I think there are some parts, some genes. Goes wrong. So when you're replicating, it's not an exact process, right? In the sense that you do make errors. But there are some parts that code for that. I'll look it up. In the sense I have some information, but